Thanks everyone for taking the time to join us for today's Cure Cancer's Researchers Lunch and Learn with our brilliant researcher, Dr. Chen Li, who's based at Monash University. This session will last around 45 minutes, and if you have any questions, please do type them into that chat box that you see there on the screen, and I'll put them into my conversation with Chen as we go through the session. If you tune in from Facebook, you might see a link to the donations in the comments section or a QR code there. This is in recognition of Breast Cancer Awareness Month, which is October. And we're very delighted to say that a generous supporter, who's requested to remain anonymous, has pledged to match any donations up to $10,000 for breast cancer this month. So that means your gift will have twice the impact. A donation of $50 will be matched to become $100, providing additional resources for cancer research. So please, if you can, do think about giving a donation via that QR code or the donation link. Also, if you haven't already, please like and follow our page to stay updated on our Lunch and Learn sessions. Our goal is to raise awareness about the critical need for cancer research funding, and your support means a lot to us. These small gestures can make a big difference in keeping the conversation going and driving progress towards a cure. So today, we had the pleasure of having one of the brightest emerging cancer researchers in Australia tell us about his work on triple-negative breast cancer. Dr. Chen Li is to share his insights in his pioneering work on an aggressive breast cancer that has, sadly, a poor response rate to current immunotherapies. You'll hear firsthand about the potential impact of this project and the life-changing difference your support is making. Immunotherapy is one of the most effective treatments against cancer, but unfortunately, it is still highly dependent on each patient's immune responses. We're here to learn more about Dr. Chen's approach in this research area, what goes on behind the scenes of the project, and also what he hopes to achieve. Dr. Chen Li, welcome.
Thanks, Nikki, for having me.
It's an absolute pleasure. So why don't we start right at the beginning, before your career in cancer research? Do you mind just sharing a bit about yourself and your interests?
Yeah, sure. Good afternoon, everyone. So I'm Chen Li. I'm from Monash Biomedicine Discovery Institute, Monash University. So it's my pleasure to talk about my research project with you and introduce my life as well. So I'm from China. So I did my undergraduate and master's in China, and then I finished my PhD here at Monash University. So my research background is actually bioinformatics or system biologies, where I can use my computational skills to mine and analyze biological data. But I think, in order to understand the biological questions significantly, I have to step into the lab and get my hands dirty so that I can better understand what's going on behind the experiment, and then we can understand the question in a more precise way.
Thank you. So we'll delve into bioinformatics in a second, but why did you become interested in cancer research as a career?
So basically, as basically all you know, I mean, cancer is kind of a most fatal disease. It's a kind of a set of so many diseases together, so many different types of cancers together, and it kills millions of people every year. I think, well, we have very advanced technologies, and we are developing new technologies every day in this area, but it's still lagging if we talk about the treatment, and it is our mission actually to leverage these new technologies to understand what's going on behind the cancers, and then propose more accurate, personalized, effective treatment to treat different types of cancers. And compared to other diseases, cancer has its own features, right? It can be really chronic, but it can be really fatal, and it can be really it can reoccur, like even though like you have finished your chemo or radiotherapies. So it significantly affects people's lifespan. So I think I need to do something that is significant. So that's why I started to be interested in cancer, and I found that there are so many things to do in developing cancer treatment. So that's why I stepped into this area, and I can say that I don't regret that. I enjoy doing this kind of research a lot.
Yeah, absolutely. It's a very important area of research. Thank you, Chen. So bioinformaticians like yourself combine computer science into biology and essentially analyze large data sets. So could you talk about this, how what you do is different from what we might know as a typical researcher?
Yeah, so basically, when everyone asks me, Chen, what bioinformatics means, it sounds really high level where then we don't really understand what it is. So basically, that's how I basically explain it to my friends. Like, look, it's bio plus informatics, so it's quite straightforward from the word itself. But we are currently using like lots of like machine learning or artificial intelligence technologies to mine the biological data. And we are able to use these artificial intelligence or machine learning techniques to predict a lot of things in biomedical data. For example, some important pathways, some important interactions, molecular interactions, so that we can provide a relatively confident or reliable list for the biologists to experiment, to verify in the lab. So that's what basically bioinformatics is. But now what I'm doing is a little bit different. Like, we are using it as a bioinformatician, I basically use data set, but I'm now trying to generate data sets by myself by doing the experiment in the lab so that I can understand how the experiment is conducted, how the data set can be generated, and how this is important for me to understand the biological questions more precisely.
Thank you. I think that explains that very well. So we at Cure Cancer have funded multiple bioinformaticians. I think last year we funded 3 of them. So we really do appreciate the importance of bioinformaticians. A silly question, but do you use a normal computer? How, if you're analyzing large data sets, what does the technology look like that you do this on?
I personally have 3 computers, if that doesn't surprise you. So basically, I have a working iMac, which is the big 27 inches Mac in my office, and I also have a Mac PC, which we're currently using at the moment. I also have a Windows laptop. And in the meantime, I also have Monash has also big high performance computing classes that I can use to perform like time consuming and computational resource consuming computation tasks. So that's all I have. That's all the toys I have for the research. Yeah.
And when you say that you look at big data sets, what numbers are we talking here?
It can be small, it can be large. So the largest one that I have dealt with, I think it has basically 5 million samples.
Wow.
So in terms of protein, 5 million proteins. So this can be super, super huge. But without actually stepping into the lab, I had no idea how difficult these data sets can be generated. It's actually millions of hours of work, millions of dollars investments. So this is really significant.
Yeah, absolutely. So you work on triple negative breast cancer, which is sadly one of the deadliest types of breast cancer you can get.
Yeah.
Can you talk a bit, please, about what your work is there and how you use bioinformatics to help with research?
So triple negative breast cancer is one of the most aggressive type of cancers in oh, sorry, I don't know what is the kind of a just because I haven't moved a little bit, just because the light went out a little bit. I'm sorry for that. So I think it's because it's one of the actually very aggressive, very fatal type of a breast cancer, right? So basically 15% of the breast cancer cases actually are triple negative breast cancers. So it's quite significant. It's a significant proportion in terms of a total diagnosed cases of breast cancer. And the reason that it's unique is because compared to other types of breast cancers, it doesn't really have 3 signature proteins. That's why we call it triple negative because we can't really detect them in the samples, right? And it's highly invasive, and it has a great tendency to for recurrence, even though basically after like major treatment which are available now, so basically chemo and radiotherapies, a lot of people, they can have their cancer back within 5 years. So it's quite quite sad truth. And what is even sadder is actually the chemo and radiotherapies are main options. Well, maybe you all know that the molecular therapy or immunotherapy is actually performing really well in different type of cancers, but unfortunately not in triple negative breast cancer. So that's why I think, yeah, we can do something about it, because our lab has a really good track record in immunotherapy development in different type of cancers. And now we are targeting breast cancer, triple negative breast cancer. And I think it's a really, really nice opportunity for me to get on board. This is really, really good opportunity. So here, basically in this project, we use both computational methods, which is bioinformatics method, coupled with experimental methods. So we're going to generate data set from the lab, and then we're going to use some methods that I published or some methods that exist in the literature to analyze the data and to find significant interest targets, protein complexes that we've been talking about in the project, to stratify patients to see whether the patient can be responsive in certain of drugs, so that we can find a well-defined molecular targets that can be actionable molecular targets that can be tested in the future in clinical trials.
So let's just go back a step with that. So you've got these 3 proteins that you talk about. And then when you're doing your analysis, are you looking at individual cases, or with these proteins and the molecular makeup of them, are you looking at trends across multiple different samples so that, as you say, you can use those for a clinical trial?
So basically what we're doing now, because it's kind of we just set up the workflow now. So what we're doing now is we're looking at the cell lines in the lab. Cell lines are easy to raise, easy to culture in the lab. So we want to do the experiment and we see what's molecular, like macro molecular targets actually are changing within the cells, because we found that we can make some we can treat the cancer cell with interferon gamma, which is kind of a cytokines in our body that can trigger inflammatory environments, right? And we are testing it. We see a significant changes in terms of the signal peptides presenting to the immune system, and we found that this kind of immune system, this kind of a changed, stimulated peptides, maybe some of them can trigger T-cell immune system, and then it can let the system know that something wrong with me, and you have to come here and take some actions. But we don't know what's actually happening within the cell and why these peptides, these signal peptides, can be triggered. The presentation of these peptides can be triggered. So we are looking at a whole set, a landscape of these macro molecular targets, molecular macromolecules in the cells that can trigger this type of a difference by using our cutting edge workflow in the lab.
Lovely, thank you. I'm just going to pause there a second. I see we've got a few new guests that have come on board. Welcome to Cure Cancer's Lunch and Learn here this afternoon with Dr. Chen Li from Monash University, and we're talking about his very important work with bioinformatics on triple negative breast cancer. So Chen, when you talk about peptides, can you explain what those are for those of us that aren't in the research world?
So yeah, so basically every time I explain to my friend, it's just use a metaphor, right? So basically, let's imagine, so when you walk in the suburb, you see a lot of houses and you walk on the road, right? So let's say if each house is a cell, right? And the roads are basically the veins, that the blood vessels, the veins that, you know, in our body. So let's say if you're, unfortunately, one house is hijacked, right, by someone,
Yeah.
And you can't really see anything from outside the house because it's super quiet, but you have no idea what's happening in the house. So if you can, for example, let's say the police car is recruiting actually on the roads, that's to see everything's well or not, but they can't see anything they can't see anything happening in your house. So if you can send some message outside your house to the police, and the police will be able to know that, okay, something happening with the house, and we have to do something. So this is similar in cancer environment. So when the normal cells becoming cancerous, something wrong happening in the cell. The cell usually fails to tell the immune system, the T cells, which is the police car basically recruiting on the road. So when finally the police is able to know that something wrong with the house, it's usually very, very late. So here, the peptides is actually the message. I would regard the peptides as the actual message that the house or the cell sending to outside. So if the cell is able to send this outside to the police or to the T cell system, the T cell can take some actions and then can kill the cell in a very early stage. But if you can't send anything outside, the cell just become more and more cancerous. It then can affect other cells, and the cancer just basically happens. So here, the peptides play a very important role in sending signals to the T cell system. And we actually, in our experiment, we found that we set the cell in a filamentary environment treated by interferon gamma or cytokines, it can actually send more signals, more peptides to the T cell system.
Wow.
But the problem is that we have no idea how the peptides were stimulated basically in the cell. We have totally no idea. This is exactly what we want to know. Because as long as we know what's happening in the cell, we can manipulate this. We can investigate. Investigate on this macromolecular targets in the cell, and we'll be able to know that how to use them for better cancer treatment. So that's basically the purpose.
Absolutely. So thank you for the analogy, first of all. That
I hope it works.
Explain something very complex very simply. So I love that analogy. Thank you. So essentially, we've got some cells that are sending messages or peptides, others are not, and obviously, if the body doesn't know, the body can't fight. So you're looking at ways that how we stimulate that messaging, so we've got more messages, so the body can fight the cancer cell as
exactly.
Well. Wonderful. So your project this year that Cure Cancer and our generous donors are funding is looking at this peptide. So what are you hoping to achieve by identifying? Is it just why they get stimulated and send messages, or do you go beyond that onto the next step of treatment as well?
So we already published a paper about destimulated messages or destimulated peptides already. So what we want to know is we want to go a little bit further to see what's the molecular mechanism behind that. So that's why we set up this fancy mass spectrometry-based workflow to identify these big proteins and their complexes that can contribute to this stimulated antigen peptide presentation to the cell surface. With this, we can hope that we will be able to know that what kind of a macromolecular targets or protein complexes can play important role in presenting more peptides to T cell system to help the cell to present more peptides to the immune system, so that we can use this to develop more effective, potentially to develop a more effective molecular immunotherapy for triple negative breast cancer.
Makes sense. Thank you. So we talked a bit about immunotherapy as well, and the important role it now plays as a relatively new treatment in cancer. For those that aren't familiar, can you touch on immunotherapy and what it is?
Yeah, so well, I'm not a huge expert of immunotherapy and all these clinical trials, but based on my understanding, I think immunotherapy is somehow to stimulate your own immune system, right? To make it effective, because for most of the cancers, the reason that we die from cancer is because the immune system just basically does nothing. When it's realized it needs to do something, it's usually very late. So it's kind of at the late stage. And this is also why for a lot of cancer types, like during the beginning stage, in the very early stage, we can't feel anything. So we think it's perfectly normal. But when we feel something, like we get sick, we got symptoms, it's usually late already. So based on my understanding, I think the aim of immunotherapy is to trigger your immune system to take actions on these cancer cells. You have to do something. You're just telling your body, you need to do something. There's something wrong with my body. So I think that's a basic idea, basic purpose. And immunotherapy basically now has been relatively promising at the moment, because we've seen a lot of clinical trials, the successful clinical trials even in melanoma, for example, and in some other cancer types. As I said, but unfortunately not in triple negative breast cancer. We've seen some like actionable targets that have been reported in different papers, but I can't really see their further investigation in the clinical trials, which is very unfortunate. So that is actually one of the hope that long-term hope that I have for doing this project.
Thank you. I've just got a couple of questions from the audience. Just a reminder for those that have joined us, if you do have questions, please pop them into the comments section there, and I'll weave them into our conversation. So a question really about the data that you use. Where do you get the data from? Is it Australian data or is it international?
So when we talk about science, we do talk about countries. We just talk about human in general. So basically, we do have some there are basically two categories, I guess. So one is the published data sets. So published data sets is contributed by different groups from every corner of the world. It can be from Europe, can be from US, it can be from China even. So they publish their paper at the same time they publish their data sets. So that's why we're able to use their data set for our own purpose. Another category is actually the private data sets. So for using the private data sets, usually needs to be cautious, because there's can be IP problem, there can be some privacy, some confidentiality problems. But usually for example, in our lab, we have our own in-house data set that hasn't been published, but it's still okay that we can use them to investigate and to get more ideas and get to know how we're going to proceed with different directions. So we don't talk about a data set from a certain country. We talk about public, private. But I know that in some other disciplines, for example, epidemiology, is probably people talk about data set from different countries, different background, people from different background. But in biomedicine, I think it's more public or private.
And then obviously you were talking about how you're looking at the proteins and the stimulation of proteins in triple negative breast cancer. Are you looking for a trend in the data? What makes you look at it and go, oh yes, this is something here that we need to explore further?
So basically we're looking at, so we have two conditions. So we have a treated and untreated, right? So treated, what I mean is here the interferon gamma, which can cause inflammatory environments for the cancer cells. So we're going to compare what protein complexes, what macromolecules actually being disturbed by the treatment of interferon gamma, because this treatment can significantly increase the peptides, if you can remember, the messages sent out to the immune system. So we want to see what's the machinery, what's the mechanism is changing in the different across treated and untreated cells. That's how we're going to define the landscape of the interrupted or changed, perturbed macro molecules in treated and untreated situations. That's what we're going to look at. That's a very basic question we need to answer. And after we identify these protein complexes, these targets, we can then investigate how to use them into development of molecular therapy or immunotherapies in triple-negative breast cancer.
Yeah, so thank you. That explains that well. So that was the next question. So you've identified something that you think is worth pursuing more.
Mm-hmm.
How does it transition to the clinic, or in a trial? What's the step after that?
There's actually a lot of steps. So that's why you say clinical trials usually takes a long time, because we need to, first of all, we need to verify we find the targets, and next step will be verifying the targets, right? We use independent tests to see whether this target is still valid, but can be validated, and then we will try to, if we have a lot of like collaborations with the hospital, so we next step, we're gonna look at these targets in biopsies. That's kind of very important, right? So that's how. Oh, I'm pretty sure there are many additional steps in between. So this kind of a really lengthy progress, but I'm pretty sure that a lot of people are working on this. So that's how it's usually costly and very lengthy procedure, unfortunately.
Look, thank you. So for those who have joined, we have the pleasure of talking to Dr. Chen Li for probably the next 10 minutes or so about his wonderful work in triple-negative breast cancer. So, Chen, if we talk about your day-to-day research, are you at Monash University there in the labs, or are you at home? How do you work? And how's the team set up?
So basically, I used to, well, as a bioinformatician, I'm totally flexible because I can work at home, so it doesn't affect anything. But since this year, because I got supported by this fancy grant, I need to work in the lab, because I really want to use this opportunity to set up the workflow, because it's quite unique in Australia, as I can say. It's quite unique workflow, and we need to invest significant amount of time in setting up this workflow. So I have to work in the lab. For example, when I was trying to raise my triple-negative breast cancer cells in the lab, I basically spent 2 months doing that. So it's quite a significant amount of time you need to investigate. You need to invest and investigate as well. So my team is quite so we have a student here. We have a very experienced PhD students that are helping me with all this setup of this workflow. We also have collaborators from Australia and from Denmark as well, who used to work on this workflow. So we are currently collaborating with them to set up this wonderful workflow. And I also have some bioinformatician collaborators, who I'm going to work with on analyzing the data set after the experiment is done. So it's quite multidisciplinary team with people from all over the world. It's really, I think, thanks to this grant, I was able to set up a multidisciplinary team that contains biologists, scientists, bioinformaticians from different countries. It's quite a nice opportunity.
I mean, that sounds phenomenal, and obviously that's a core focus of our funding at Cure Cancer, to fund innovation and kickstart new ideas. So when you talk about this being a new idea or something that's different, why has it not been thought of or done in Australia before?
So yeah, so what I'm talking about is actually the workflow, or the things that we're going to investigate, because before the Cure Cancer grant, I got a HMRC research fellow grant. So basically, I went to Switzerland basically for 2 years, where I started this workflow, this mass spectrometry based workflow for protein complex identification. I realized this can be really helpful, because it can apply to anything, like any disease, including triple-negative breast cancer. So I really want to bring this workflow back to Australia and to contribute to Australian health biomedical research. So that's the key, mindset that I have. So this workflow is kind of unique it's very effective and it's very kind of unique in the protein complex identification. And we have so many top tier publications describing this in recent years, which is a testament of how important this technology is. So now I'm able to, because of the grant, I'm able to set up this very fantastic workflow here in Australia. And we were able to, like say, if it's successful, we'll be able to use this to apply this to different type of diseases and cancer types to facilitate or further advance the cancer related research.
Well, that's incredible, and to be able to use it across multiple cancer types as well. Thank you for your work. Why did you start then looking at triple negative breast cancer with this workflow rather than another cancer type?
Well, one thing is that it's very invasive, right? It's kind of really invasive and had a great tendency to be for the occurrence. So it's very hard to fight type of cancer. That's why I think I should target this challenging cancer type. I love challenges. This is one thing, and the other thing is that we have a colleague, previously the colleague whose grandma actually died from this as well, triple negative breast cancer. And I know that because I searched online a little bit, I found that this is really prevalent, because I said that 50% of breast cancer cases actually are triple negative breast cancer. And also I've been in different sort of consumer supporting groups like on Facebook and Twitter. I saw that sad news every day, and I saw people going through very suffering chemo and radiotherapies, and they are just updating basically their progress every day in the group. And it's very sad to see that some people lose or lost their fight to the cancer. So I think, well, why not? We have the technologies. We have both computational experiment technologies. It is our responsibility to do something, at least to do something to further advance the possibility of developing effective immunotherapies for this type of invasive and evil type of cancer. So that's my motivation to work on this triple negative breast cancer.
Well, what a wonderful motivation, and I'm sure everyone on the call appreciates you and your team's work. So thank you, Chen. We touched on the funding, and as many of our supporters know, it is difficult to get a lot of funding, particularly when you're a young emerging researcher and don't have a long history of research behind you. What did the Cure Cancer grant funding mean for you to be able to start this unique project?
So because of the grant, I think I'm able to take a big jump now, because it's kind of a career shifting, or it's a mindset shifting opportunity. Because as a bioinformatician who has been working in this area for like more than 10 years, right, it's getting harder and harder for you to jump out of your comfort zone and to do something that's totally strange to you. Even though I'm working working closely with biologists, oncologists in this area, but it's actually the first time for me to step into the lab, get used to all this experiment environment, and to generate data sets, to raise the cancer cells and do the analysis by myself. So this is one of just one of a kind opportunity for me to do this. So this is what it brings me. So that's why I think, because of this, I can confidently say that I can also do some experiment, not in a very sophisticated way, but I can do something. I can now understand what's happening. So this is actually very, very helpful, very, very inspirational in my career development.
Thank you. So what are your hopes for the future? You talked about obviously wanting to identify why these proteins, or what enables them to send messages, and look at taking that to clinical trials through hundreds of steps, but to get there and ultimately get there. What's your big hope for the future with the research that you are doing?
I'm a dreamer, so but not daydreamer, more realistic dreamer. I hope that my ultimate hope is that cancer can be a curable disease. You know, it doesn't mean that you get cancer, you're gonna die. This is my ultimate hope, because cancer basically happens every day, but the most important thing is that we can make it treatable. So next time when you're diagnosed with cancer, that's fine. Just you'll be treated anyway. So that's fine. Don't worry about that. That's my ultimate hope. But I'm also a realistic person. So what I totally understand that in order to achieve that, there are millions of steps to achieve. So I would like to, based on this program, I would like to, and just to make sure that I can identify these interesting targets, to provide these interesting targets to the research community, and see, okay, we can further advance the triple-negative breast cancer immunotherapy by using these targets, using the findings, using the discoveries by this grant. That's actually very, very, I would be super happy with that. And I hope that I can keep contributing to this area and to make it closer and closer to be treatable disease. So that's actually my kind of a short-term, long-term hope, I guess.
Yeah, absolutely. Well, thank you for sharing that. And before we wrap up, do you have any last words? We've got obviously lots of donors and supporters here whose generous donations have helped go towards your funding. Do you have any last words that you'd like to share with our donors?
Yeah, so maybe I can say something on behalf of all the young and early career scientists in Australia. So, unfortunately, because of the pandemic, because of the economic situation, like more and more talented biologists or bioinformaticians, they have to change their career. They have to step out of academia, which is quite sad, because you have to realize that it is the effort from these millions of talented scientists that now we have further advanced the technologies, we have further advanced the treatment of so many diseases, including cancers. So I would encourage you, I would urge you to do whatever you can to help us keep doing this exciting experiments to make some exciting discoveries in disease treatment, especially in cancer treatment. Together we can fight, but we need your help, and I think with your help we were able to keep this going, and we can achieve our long-term goal. Thank you.
I couldn't have said that better myself. Well, thank you. It's been an absolute delight. I know I've enjoyed, and I'm sure our audience has enjoyed listening to your work. Dr. Chen Li, thank you on behalf of everyone for the work you're doing.
Thank you.
It's a delight, and we very much look forward to hearing how you progress and the results as you work through your workflow as well.
Thank you.
Thank you. So thank you to you all for joining us today. We will, of course, put this on our website and also send this out via email to you. Please, as we talked about before, there is a QR code there. We are matching donations for the month of October because it is Breast Cancer Awareness Month. So if you are able to make any donation, it will be doubled and the impact will be doubled. So thank you for those that can. Please join us at the same time on the 26th of October for our Lunch and Learn with Dr. Eric Kusnadi. Eric is at the Peter MacCallum Cancer Centre in Melbourne, and we'll be talking about his very important work on prostate cancer. But until then, thank you for joining us. Stay happy and healthy, and we'll see you on the 26th.
Thank you.